Now I'm on the clock here. So let me just share my screen here. Hopefully we'll be able to get a not presenter view. Is that coming in all right?
Yep, that looks good on my end.
Cool. All right. Well, thanks everyone for having me here. I'm excited to talk to you. My project, I guess I've loosely titled Fitness and Functional Traits, but it's really taking a broad approach with demography to answer a lot of ecological questions. So this view you can see here, the picture in the background, that's from the Miriam Powell Research Station. Many of you are probably familiar. I really like giving this talk to the people who actually know the study system. So it's very cool that you guys are going to be able to relate a lot to these pictures. And when I say where we're talking about, you'll actually be able to think about having been there. So for the outline, I'm really going to divide this into 3 parts. The first and biggest portion of things is going to be the local study that takes place around Flagstaff. That's the Hill Wild Bill Demography Study. It's an observational study that's been going on for 20 years. It's 20 plus years at this point. Really, Margaret Moore has been the one, you guys probably know her, has been carrying the torch on that and keeping things rolling, so we have an awesome data set to work with. The second thing I'll touch on briefly is the GRAMP studies. This is an experimental approach. It's kind of adjacent to what we're doing in the Hill Wild Bill study, except it incorporates a more experimental design with precipitation manipulation and is outside of the study system where Hill Wild Bill takes place. And then I also like to talk about some broader applications of some of the techniques I'll be introducing during the talk. Or like, as I like to think of it, using demography in fun ways that we can answer other questions. So to hit you with 2 overview slides in a row, this is the first part of the talk where I'm going to give some background, particularly in regard to fitness and functional traits. So one way is how do we measure fitness, and how am I going to talk about it in this study? What are functional traits and what they mean, and some definitions of them, and how these 2 are fundamentally related to each other. Then I'll give you a little bit of background on the study design objectives for the Hill Wild Bill demography, and then how we're going to model some of the trait environment relationships. I'll report some of the population growth estimates we have for some of the species that we've been studying, and then outline some of the cool interactions that we've seen between traits and environment that really kind of make us excited for the future. Speaking of the future, the things we'd like to do to enhance our models are going to be also discussed, as well as how this ties in with the experimental approaches that we're going to be taking in the GRAMP studies. So fitness in general, there's many different definitions of fitness that kind of vary based on which subfield of ecology you're going on, whether it's the probability of passing on your genes to the next generation, an individual's lifetime reproductive output, relative success to other individuals in the community. All these are valid, but for this talk, we're going to consider our metric of fitness is the population growth rate, also known as lambda. So at a population level, how well is this population performing? We're really interested in using this as our metric of fitness because it allows us to evaluate the persistence and or growth of populations. So if something's got a positive growth rate, or if it's actually declining and maybe heading towards extirpation. So that fitness is derived from vital rates or growth, survival, reproduction. And when we say lambda, 1 is kind of our reference point. So a lambda of less than 1 indicates a population is in decline, where lambda greater than 1 it indicates that population is growing. So a lambda value of 1.1 is roughly equivalent to saying that the population is growing at a rate of 10%. So vital rates and fitness here. Again, I said fitness, as we consider it, this population growth rate is a function of demographic parameters. We consider those growth, survival, and reproduction. And there's well-documented trade-offs in these vital rates. So for a specific species or population, individuals in there may respond to their environment by surviving better, but reducing fecundity. So those are kind of some fundamental trade-offs that we see at the level of these demographic parameters or vital rates. So like I said, simultaneous changes in these vital rates are kind of what we would call different strategies, can make fitness difficult to calculate because it obscures the net effect of fitness. So a change in vital rates is not always equivalent to a change in fitness. So you can see here, while a species may increase its growth while simultaneously reducing its survival, that leaves the fitness or lambda values roughly unchanged. And here there's many different growth, survival, reproduction strategies that persist in this triangular space, and species can kind of modify their vital rates and still be successful. All these may produce a similar fitness value while having slightly different measures of growth, survival, or reproduction. So changes in trait values or functional traits, which we'll get into later, may influence multiple vital rates. So if a functional trait that's important, or how a characteristic of an individual that makes it maybe survive better, or in this case, as illustrated in this graph, worse, may help it grow a little faster and be more fecund. So when considering how a trait impacts these vital rates, we nearly need to consider a holistic viewpoint to evaluate what the net effect of fitness is when we're talking about a trait-based perspective. So a little bit about what functional traits are. They're characteristics of a plant that we can measure, and it's related to how it interacts with the environment. So phenology, physiology, morphology, all these things are characteristics of a plant that we can measure between species and compare populations using this way. So two traits that I just wanted to introduce quickly here to give you an idea of how that would work in an environmental context are SLA, which is specific leaf area. That's typically higher values of specific leaf area are typically associated with resource acquisition. There's also leaf dry matter content. Higher values of leaf dry matter content are typically associated with drought tolerance or survivability. Now we can measure SLA or LDMC on any plant organism. It doesn't matter if it's a graminoid or a tree. Some of them are tougher, like succulents, but we can take these measurements from any individual and use that as kind of our comparison. So we're leaving our phylogeny in the background using traits to make predictions here. So along a resource availability gradient, what we'd kind of expect to see is lower values of SLA and higher values of leaf dry matter content. That'd be consistent with a survive and maintain type of strategy, where when you have a high values of high resource availability, maybe a strategy that's more resource hungry, so higher values of SLA and lower values of LDMC might be more successful. So this is in the forestry building here, where we've got a lot of these individuals that were collected after a hard day's field work that we're going to be taking a lot of these trait measurements from after rehydrating them and getting them ready to go into the lab. So we always consider I always like to consider these functional trait values important because the species persistence in an environment can often be constrained by its traits. This is a process known as environmental filtering. And one of the fundamental tenets of trait-based ecology is that variation in traits is going to happen. There's always variation. No individuals are exactly alike, but interspecific variation in traits is always going to outweigh intra-specific variation in traits. So ultimately, organisms are bound by their biology and will not be able to express beyond certain values. However, they will be able to express a range, and there is some type of plastic response that you see in functional trait values. So over here, this is how environmental filtering from a trait-based perspective would work. There is a certain value of a functional trait that may be that occurs along this environmental gradient, and that value, a certain value of that trait will be more successful than others. Now we see trait distributions here for all individuals that are in what we call maybe a regional species pool. And under these certain environmental conditions, only a few species are able to persist based on their trait values and the distributions of the trait values that they inherently possess. We believe that this predicted relative abundance of species will reflect the distribution of trait values based on the particular environmental filter that we see. So to give an example that maybe everyone is a little more familiar with, especially along elevational gradients. Bark thickness is one of those traits that we consider functional, and it's very adaptive in some situations. So when we look at it from how this affects vital rates, so thick bark may help trees survive fires. That will increase the survival. However, it demands a large amount of resources to develop that thick bark, and that might otherwise be spent on growth. So this is data from Pete et al. In 1978, where it's from the Front Range region of the Rocky Mountains. Ponderosa pine is really prevalent there, just like it is out in Arizona at lower elevations. Fire is extremely prevalent. But as you increase elevation, bark thickness as a community-weighted mean value steadily decreases. Very strong relationship here. And we take that to be because there's an adaptive value of bark thickness that is dependent on the environmental context. The environmental context here in particular is the prevalence of fire. So when we have fire, bark thickness values will increase, or thick bark values will increase your fitness. While when fire is less prevalent, fitness values are expected to decrease. That's why, you know, we see ponderosa pine at low elevations are frequently burned. But as you head higher up into mixed conifer where fires are not as prevalent, ponderosa pine isn't anywhere to be found. Well, not as much, especially not in the Rocky Mountains, but out where you guys are probably is. So putting these next to each other, we see that elevation or the elevation or prevalence of fire is our environmental gradient, or how the filter. And then this functional trait, which we have as bark thickness, can dictate the species distributions throughout the study zone. So what we actually did, this might be familiar to you guys. It's kind of north of Flagstaff. This is at one of our study areas called Reese Tank. So maybe a little bit dense here compared to the historic conditions, but ponderosa pine cover. And then really what we're going to be studying is these grasses that persist all throughout. Perennial Gramanoids are the primary focus of this study. Particularly these 10 of them here. So all these 10 Gramanoids are very important in the understory out near Flagstaff. These are the 10 most dominant ones. There's some different photosynthetic pathways. This is actually Muhlenbergia tricalypsis, blue grama, white mountain sedge, Arizona fescue. You guys are probably familiar with a lot of these populations. So what we did was we visited all these populations and got trait values for each of them. So for each species at each site, we took collections and measured all these different characteristics, specifically here at LDMC. Last summer, we spent a lot of time gathering root trait data for them, because above ground and below ground energy economics are often very different things. So it's good to have, again, a whole organism point of view when you're talking about functional traits. And we're pairing this with demographic data for each species within each quadrat. I'll get that a little bit, but it's important that we have this demographic data so we can ultimately develop population models, or these IPMs, so we can evaluate how populations of individual species are doing at each site along an environmental gradient. And we also wanted to identify traits that we measured that predict the plant performance across this environmental gradient. So that's linking traits to fitness is kind of the core thrust of the project. All right, here. So this is a bit of the area hopefully this is familiar to a lot of you. So we've got Flagstaff over here, San Francisco Peaks. This is running down to West Flag is over here. Rogers Lake is out this way. Miriam Powell Arboretum somewhere over here. But this is a map of this is the total extent of the Hill Wild Bell study area. Each of these red points here represents one of those individual quadrats that I'll show you a picture of later, where we've visited, or not me personally, but a lot of people have visited for over 20 years to get this demographic data. And zooming in a little bit, each of these each of these quadrats or red dots is roughly organized into study sites. So government knolls is a collection of sites out here. There's 6 quadrats contained within this study area. And then zooming in a little farther than that, we can now see, put this a little better into perspective, where we have individual pine trees, and then all of our quadrats where we collect our demographic data from. So these are 1 meter by 1 meter quadrats. They look a lot like this. These are just old angle irons that I believe were put out. Mario will have to give me the exact date, but it was early in the 20th century by some very famous ecologists. And my advisor, Daniel Laughlin, went out and unearthed a lot of these things at one point, which I think is really cool. But each year, these are surveyed, and this is how this is a field collection data sheet. So each one of these individuals is, depending if it's a graminoid or an annual form, is traced to give you polygons of that individual, so we can get estimates of its basal area. We can track these individuals through a year and get estimates of growth and survival. And then using recruitment for individuals that didn't occur last year, we can get estimates of reproduction as well. So what that looks like once we've taken these sheets back to the lab and someone has diligently digitized them is this. So each of these individual points here represents a species that we track. In this case, Bouteloua gracilis is all species with this red hatching on it. And what I'll do is I'll show you this quick little video of one quadrat throughout, I think it's 10, 15 years that we've got data for. So you can see as each one of these changes, this is an annual transition. And you can see there's some variation in where a polygon is, but that ultimately allows us to track individuals to get an idea of who's growing, who's surviving, and what species are reproducing. So that's a long time to have demographic data like this, 20 years, it's pretty incredible. So we're very fortunate to be working with this data set. So what the overall goal of the study is to describe the fitness for these species and calculate these population growth rates using a tool called an integral projection model. This is a fancy new math way to kind of calculate how populations are doing. We'll get a little bit more into it later. And then use the model of fitness using traits and environment. So what we're looking for is an interactive effect between traits and environment that we would expect to see when there's an environmental filtering taking place. So if we can find which species are doing well under certain environmental contexts, we expect that due to the trade-offs that we hypothesize to observe, they might not be doing as well under different environmental situations. So, so this is when I say population models, integral projection models, this is kind of the net output for what that is. So lambda values for all of our graminoid species range from 0.7 to 0.12. So that would be roughly a 30% decline to a 20% growth. Those are not, I would not consider those truth. I doubt that there's anything that's declining at a rate of 30% that's not going to go extirpate very quickly. However, the real takeaway is that it's either plus or minus 1. Are they doing well or are they not? And I've seen other studies published where they report, in some cases, double-digit lambda values. And so I'm extremely happy that our results fall within what I would consider a pretty reasonable range, at least for the values. And now I just want to take a time to orient everyone on this. This is an IPM kernel. So for this is Muhlenbergia, Montana, one of our low precipitation sites, Big Phil, which is actually almost in Flag city limits. I believe it's just southeast of the golf course out there. It's pretty close by. You're driving on a paved road right up to the site there. But the way to interpret this figure here, so this is a 1-to-1 line, the black line that I've laid on here. And the values here are transition probabilities. So we've got them on a log scale for the size of each individual at year t, and this is the size at year t plus 1. So IPM is divided fundamentally into 2 parts. We have the growth survival kernel. So these probabilities are a function of an individual's growth and their probability of survival. So you can see that individuals over here are like the small individuals have a very high probability of growing. They're above this 1-to-1 line. And it's also a function of survival as well. So it's very likely that the individuals will survive and grow up to a point right here, which is roughly where the they'd fall below the 1-to-1 line where they would start dying. But now they incorporate the second part, which is the reproduction kernel. So this is basically individuals of these size classes are reintroducing offspring or recruits into the system at these probability rates. So you can kind of broadly see the two different components of an IPM kernel. And this will vary based on which species and which site, or so which population we're looking at. So I think we found that the lambda values differ with trait and environment. So really that's kind of one of the core things that we were hoping to see from our study, was that there was an interactive effect between traits and environment. I don't have time to go into all the traits and all the environmental variables, but what we saw here, you can see lambda is kind of our response, and then one of our traits, LDMC, and then precipitation values. So this is an interaction surface for the LDMC precipitation. So this is kind of our evidence for environmental filtering and the adaptive value of traits along an environmental gradient. We're going to pull out a couple species and look at the IPM that's for each of these as well. So this is the one that we looked at originally, along with the LDMC, or the trait value that we're interested in here. So there's a high value of LDMC. You can see that these are both, their LDMC is a ratio, ranges from 0 to 1. And this is near the very high end of our range for leaf dry matter contact. So this is the top panels here are Muhlenbergia montana, again at a low precipitation site and a high precipitation site. Now we're going to look at another species, Elamis elamoides, bottlebush squirrel tail. I'm sure you guys have seen it out in the woods and flag. It's got one of the lower LDMC values from among our range, and again, some interpopulation variation. However, the difference between species is much greater than that we'd see, or the difference between species is much greater than that between populations. So let's take a look at where some of this kind of falls on the interactive or the interaction plane. So with a high value of LDMC and at a low precipitation site, we find our species occurs here. So the difference between these sites, I would say that individuals that are smaller individuals are much more likely to survive and grow when there's high precipitation available. And they're also reproducing a little bit more. So that leads to a lambda value of 1.06, which means this population is growing. Those positive conditions for that species under those environmental conditions. However, over here, we see that the biggest difference is really at these small individuals. So maybe, what we could hypothesize, one of the reasons for a lower lambda value is that those small individuals can't take the drought at this site and are not as successful. So we see that this population, again, high precipitation with the same high leaf dry matter content, will give you a high lambda value. Now looking at another species, again, same environmental, just comparing the same two sites. So just two populations of another species, that has a much lower different trait value. Now when there's low precipitation here, Elamis elamoides actually looks very successful. It looks like reproduction is happening at a much greater rate than you would see at this other population out in Kendrick panel. So this is a close, kind of just southwest of the mountains. So you get a lot of, or it's just south, I think, of Kendrick Peak. But you get a lot of precipitation out there. And so who knows why these individuals, we're hypothesizing that's the trait values that allow them to better persist under these environmental conditions. So different trait values can often lead to inverse effects of, or inverse growth and decline of populations under a wide range of environmental conditions. Again, I kind of picked the extremes here just to illustrate the point, but we do see some really great trends, and this interaction space here is from 82 different populations that we've covered through the Hill Wild Bill study area. So, and this is where this population would be on that interaction plane. So one of the things that we're also working on improving is the reproduction of evaluation. So as you can see, think about how we take that estimate of reproduction each year. We are going out and just tracing individuals that are there. We really don't have a way to evaluate if this year's seed fall is next year's recruits. So what we're trying to do is incorporate the seed bank and seed bank dynamics into our models. That'll hopefully give us a much better or much more accurate just way to describe what's happening. And persistence in the seed bank could be a very important thing for predicting how these populations will respond. Especially in an area where fire is prevalent, and out in Flagstaff, even though there's historical fire suppression, we would expect that species that are able to persist in the seed bank and maybe are opportunistically germinating after fires will get a benefit from that if we're able to model that appropriately in our reproduction kernel. Okay. So I'll go over some future objectives that we're going to touch before I conclude with the Hill Wild Bill and move on to the next part of the talk. But what we want to do is expand the environment in the models. And what I mean by that is that we really want to incorporate things like density dependence and competition models, because these plants aren't just growing, there's not the same amount of density between each of these study sites. So if we're considering things like precipitation and temperature, that's great, but that's not equivalent when there's maybe, if you're competing with a bunch of individuals of your same species, or if there's a bunch of other individuals from other species that are growing very close by who are competing for those resources. Maybe that water is very important, a limiting resource there. Maybe some other species are more capable at extracting nitrogen. So expanding the environmental value component through incorporating density dependence and modeling other species occurrence that occur near these individual polygons is important to do. Also, move farther to evaluate which traits drive the population fitness in different environmental conditions. So that's just building more of these trait environment models to see what is the best predictor. What can we measured a lot of stuff, but what can we really hang our hat on to make projections outside of the study system, which is the next thing that we really want to do. You can build a model on data, but it's really not useful unless you're using it to predict something, or so you can forecast the future or you can forecast outside your system. The next thing that I'll talk about is an experimental study, the GRAMPS study, where we're looking to take those models that we developed from the Hill Wild Bill demography and make projections about other species that are not the same species that occur in Hill Wild Bill, but see if we can forecast their population levels in response to experimental precipitation manipulation. So some takeaways from that study, the one that I've been most heavily involved with here, is that traits have a variable influence on the vital rates, and that influence is mediated by the environment. So we can't really determine the net effect of traits on fitness by considering just these individual vital rates. So really building these population models that take a real holistic approach to evaluating fitness through using integral projection models. I think that's very important and something that we need to keep in mind moving forward. So experimental validation, like I touched on, it's super important because if we can take these models and develop them in a small subset of systems and then project what might be happening under a rapidly changing climate. Are we going to see changes in species composition? Are there going to be range limit expansions or contractions? Will we be able to predict persistence or extirpation of rare or endemic species using these population models? I certainly hope so. So that's the last thoughts I have on Hill Wild Bill. And now I'd like to take a look a little bit farther. So what I set up as the experimental context for how we're going to take these models and really put them to the test experimentally. That's that's going to be the GRAMP study. So this is an experiment that's really kind of contiguous to the Hill Wild Bill study in both concept and space. Except for rather than being just a strictly observational project, there's an experimental component. So if you can see here, these are actually, that's the peaks in the distance there. Many of you recognize that view coming up or coming down 89 south towards Flagstaff. There's 5 study sites out here. This is one of our lower elevation, heavier drought sites. I think this one is Black Point, but you can see out there, they've set up these experimental rainfall catch things. So as rain comes here, it's drained off into a gutter and stored in these barrels. So that's a 50% reduction in precipitation that they're implementing there. And then the water that's stored in these barrels is then pumped back in, through these little spigots out here that you can kind of make out. So there's actually an addition of a precipitation addition component as well. So there's a control as well as a 50% reduction and a roughly 50% addition. Obviously, there's some loss when you're transferring the barrels or transferring the water from the barrels into another through the pump, but we keep track of that as well. So this is one of the lower elevations. I said desert shrubland here. We also have a desert grassland site, and it really spans the whole range of things. This is out near Camp Colton. So this is up in mixed con area, so mixed conifer. You can see the difference in productivity between the sites is immediately apparent. Camp Colton gets a lot of precipitation from the peaks, very lush out there. It's a really beautiful place to be. That's one of our field techs, Oliver, hard at work, taking down some demographic data. So I'll just kind of give you a side-by-side of these two different studies. So Hill Wild Bill here is on the left and GRAMP's experiments on the right. And for the environmental gradient, the Hill Wild Bill study is kind of constrained within most of the ponderosa pine grassland area. So there's variation obviously in precipitation, but most species are common. There's not a whole ton of species turnover. It's usually just the same species in their different proportions. Some occur and some don't. It's also observational. So one of the strengths is the temporal depth of the data set, but there's no real experimental component to it. Now, the GRAMPS has been going on for 5 years, which is also a great amount of time. Some people build population models with only like 1 or 2 annual transitions, which is kind of tough and can lead to a lot of those really extreme lambda values that I was referencing earlier. So having 5 years is great, and 5 sites along 5 totally different, forest or cover types. So all the way from desert shrubland and grassland up to mixed con. It's also, each of those sites is experimental. So there's like plus and minus precipitation treatments going on. And there's a very small overlap in species between sites. They're totally different cover types. So some extremely widely distributed species such as Futalo gracilis, blue grama, is present on more than one site. There's some overlap, but for the majority, it's a totally new ecosystem there. For those results, you have to stay tuned. That's one of the projects that we've got going on right now, is once we finish building these models from Hill Wild Bill, we want to take those and really see if we can predict how the species in the GRAMP study will respond based on their trait values. So that's the real test of our linking traits to fitness, is if we can predict that under an experimental setting. That is the lovely root grama here blooming away at Blue Chute, which is one of the intermediate elevation sites for the GRAMPS experimental system. Cool. So I'd like to talk about some broader applications of IPMs and demography in general that I think are very cool and really are leading the way for how we can answer some of these large ecological questions. the first thing that I want to introduce is these demographic distribution models. So these combine an integral projection model approach with Bayesian models to predict species distributions. So as I outlined earlier, the environmental context for a lot of these things is extremely important. And calculating the fitness or the population growth of species is dependent on the environmental context, but you can make estimates of how individual vital rates would be impacted based on widely available climate data. So by using integral projection models from a very small subset of data, we can then build or make projections of these models that where species will occur and under what conditions, based on if they have a positive or negative lambda value. That's kind of just using demographics as a response. So like I said, one of the applications, I'll cover a case study where they built one of these demographic distribution models in the Cape of South Africa. It's very cool, I think, a way that we can integrate the demographics approach. And as I've described, I started off with a very local study that's being paired with a regional study, and this is something that you can apply almost continent-wide, these demographic distribution models. Another very interesting way that they apply this is evaluating invasive species threats. So if a species is introduced into an area, they can calculate demographics along a small environmental gradient and then project that farther to potentially outline where the species may spread to if left unchecked. So that's a good way to evaluate whether a species may persist in novel areas if it's introduced. And that can really help focus management efforts. Should we really be spending limited resources targeting some of these species that may not spread or won't be successful? Or it could also highlight ones where it would be an emergency need where a lot of resources should be directed towards it. So that's an overview of these things. Going to the case study now, which is very interesting. So to really build up this method, we're using common species with known population distributions with this demographic distribution model. So to do that, you build population models on a subset of species from a small environmental gradient. So it's data that's relatively easy to collect. Then you can do things like sensitivity and elasticity analysis to evaluate how changes in growth and survival will affect overall population growth rate. Then you can, and it's using Bayesian approaches to propagate your uncertainty along each step in that process, but you can get basically an overlay of where you think species will be successful and where they won't be. So from what they did, they had a small subset of the study sites where they generated these models, and they were able to predict, they then took a bunch of surveys from the area where they were evaluating where the population may spread to, and they're about 70% accurate with predicting presence and absence at a very local site. So that's pretty good in and of itself, but really it really comes out when you look at it from a regional scale, where you can define the potential ranges of species. So I'd like to take a look at some maps here. This is down in South Africa, where we have a Proteus species, and this is not the species epithet. This is an atlas where they have presence and absence. These are known observations of this sugar bush throughout this whole region. So when they built these models using the approach I described, they were looking at lambda as their response, and basically the way that they implemented this was, okay, if a species, this is endemic, it's not, or not endemic, it's native, it's not introduced. So if we would project that the species has a growth rate or a positive growth rate there, we think it would persist. However, if the model believes that the population would decline there, we think it's unlikely that it would persist. So it's tough to make out with the topography here, but they were able to predict the presence and absence along different topography, and really outline where that species is successful, and you're likely to see it occurring. So I think that's very cool to be able to project where we might see populations that are in danger, or things that, if it's of high conservation concern, we might want to target efforts towards there. Just want to close on like some final thoughts with demography. IPMs are a very powerful tool. Being able to get information that is indicative of how a population is doing, I think is it's great to use that as a metric for how is for impact of climate change, maybe management efforts. And it's really not too hard to get data and at a local scale, and then you can make projections much beyond that at even regional or ecosystem level, depending on how well informed your models are. And some of the new modeling approaches are just incredible, and I think it's a very cool way to apply demographics. So I think population demography is kind of a fundamental unit that we should be conscious of moving forward. And the applications are broad. I just kind of talked about 3 different scales at which we are using demography, and how integral projection models can be applied in all those cases. So whether it's the Hill Wild Bill study, we've got a local focused demographic study where we're building these populations really with a focus on traits and fine scale climatic variation. Then at the more local or regional scale, where we're looking from desert shrubland all the way up to mixed conifer in the Gramp study. And then, as I described with the in South Africa, where they're really looking at a whole species range across a wide varied environment, and then potentially predicting what species are going to be invasive, what ones will be most harmful based on their spread, and how they will impact other species. I think that demography is one of the best tools that we can use. And with that, I'd like to extend a big hearty thank you, first of all, to you guys for hosting me here and for listening to talk. So many collaborators here at the University of Wyoming and there at Northern Arizona University. I'm sure some of the names are familiar. Big thank you to everyone, as well as our collaborators at UC Davis and with the USGS. And with that, I'd be happy to take some questions.
Yeah, so you guys can unmute yourselves when your hands are raised, or you can just type them in the chat and we'll read them off. Connor, I saw your, oh, that's an applause thing, never mind.
Yeah, I don't have a question. I just wanted to say I enjoyed getting the understory perspective. I feel like we probably don't hear that enough in the School of Forestry, and you had some awesome photos too. So thanks for sharing your research, Dave.
Happy to. Yeah, I was thinking probably not too often that everyone in the forestry department has the plant dichotomous key out and is measuring legules and things like that. A little easier to take a tree perspective, but I like the graminoids myself. That's a great point, Carol. Yeah, obviously the integrity of these ecosystems and is dependent on a lot of the base plants, but we do want to incorporate population models of wildlife. Would be very interesting to see paired with concurrent plant demographics.
Dave, I've got a question about oh, I'm sorry, Margaret. I just started when you raised your hand. Do you want to go?
No, I took my hand back down. I do have a couple questions or to ask Dave, but I could wait because these would be pretty specific. Well, they're C3, C4 related questions about that trait. I'll just wait, Pete.
Okay. Well, this is just a kind of broad question about the application of these kinds of models. I realize that, as you mentioned, there's not enough data to make these to make detailed demographic modeling of a whole lot of species. That's why this data set is so unique. But if we had more data, and particularly if we had data about species that seem likely to be invasive with climate change, would that be helpful in letting us predict what community composition might look like here in 20 or 40 or 50 years?
Yeah, I think even having, obviously, like you said, the strength is that we have lots of transitions and annual data here, but so much can be done even just from 1 year's worth. Dr. Corey Merrill gave a presentation, we hosted him and he gave a seminar, and there's some unpublished data that I really wish I could have shared here, where they took a small sample of an invasive species in New England, and were able to almost perfectly predict the occurrence of it from just 1 year of lambda estimates using the methods I described. So from just a small measure in New England, it's a mustard that's invasive, I think, all the way down to the Carolinas. And they were almost very clearly able to outline how it's, where you would find it. So I think that no matter, even if you have a little bit of data, I think that the findings from these things are very important, where you could project community composition changes, like you said, in 30 years from now, even just from a couple years worth of data collection. So I really do think they're incredibly powerful. And I'm
Thanks.
looking, yeah.
And so Pete, I think Ethan Tabor had a similar question. So maybe Ethan could ask his question, and then I'll follow up with my C3, C4 question.
Sure. Yeah, we'll see if it's the same question. Yeah, so I just thought it was interesting that, you saw that the Elemis lambda went up. So it was performing better in the dry site. But as a C3 species, it's kind of what I wouldn't expect that as much versus like the Muhlenbergia being a C4, it had lower lambda values in the dry site. So, I mean, generally we kind of think of C4 species doing better in drier, warmer environments. And just was going to ask if you guys have any speculation as to why that might be, or I don't know. Yeah, just found that interesting.
Sure. Yeah, that is one of the reasons I picked that LDMC relationship there and those two species was that it is totally contrary to what we would expect, even at the trait level. So as I had touched on earlier, leaf dry matter content is kind of expected to be associated, higher values are expected to be associated with better performance under drought conditions. And we saw the total inverse of that. And I'm sure that may have something to do with the C3, C4, but I think one of the reasons for that may be the range of the environment that we captured. So if you consider that when the species, like a species distribution model, if you consider that their abundance is likely to be normally distributed, maybe we only caught the part of it where it's trending downwards with our small sampling window. So that is something to consider. I don't have a direct answer yet because we'd have to build a lot more models and see if other traits are better predictors, and I'm sure there will be interactions between traits, and maybe the below ground picture will be more clear. But yeah, great observation, because it is the same thing that we thought. Like, this is totally contrary to what we would expect. But maybe that's why I trust the models. So just finding out the reason why, I guess.
Yeah, totally. And yeah, I'll talk to you in the discussion about it more, maybe.
Cool. Thanks for the question.
Yeah, thanks for the answer.
What do you think, Margaret? Did we get everything there, or do you have something else you want to talk about?
I'll go ahead and just suggest that we have been looking at Arizona fescue, which is a C3 cool season species, and mountain muley, which is a warm season species. And we have been looking at this for a couple years now with integral projection models. And I think Rachel Mitchell, it'd be Ethan's major professor over there in environmental science, she's been really earth and whatever the, sorry, earth and sustainability. And we are seeing that that opposite effect. So it is, I think we've now added temperature and precipitation to those integral projection models, and that is explaining everything. And it is the timing and the season of precipitation and these non-monsoon summers that we've had is causing mountain muley, the C4, to decline. And so we are seeing lambda values starting to drop. That is coincidental, the same time with these drought and non-monsoon periods. So even though everything's been having a long 20 years, we've been in 20 years of drought, people, y'all probably know that, mega drought. We are seeing these fine scale influences on the C3, C4. And so another paper that was published, shoot, I've forgotten what it's, just a couple years ago, it's called the Dust Bowl Paradox, where they saw the same thing across the Great Plains with the opposite response in C3 and C4 as we're seeing with our data. So Pete mentioned it's 26 years of mega drought. So our data set, even though this is, these plots have been in place for 100 years since 1912, we've only been looking at the data for the last 20, but we are now capturing it. It's pretty obvious in the data. I'm kind of excited to get that publication out. But we'll talk more when we actually will meet you again there, Dave, in a few weeks, so we can talk about this more.
Oh yeah, hoping to have some much more refined models to show you guys, but it's been exciting so far because we're really making hay right now.
Yeah, the Dust Bowl Paradox, though, folks, you look it up, that paper is really good, and it's explaining really similar to what we are seeing. I'll try to send that to people.
Sure.
If
It's
they're
Alan Knapp and Mindy Smith.
Yeah.
Yep.
Alan Knapp, who was at, he's just a famous grassland ecologist that was there in wherever he was, and now is at Colorado State. And then one of the people there in ECOS was on that same paper. And I can't remember his name either. But all right, well, I'll share that if we find it. Thanks.
Looks like Pete just linked that article to the chat. Are there any other questions for Dave? All right, well, if not, maybe we can switch over to the post-seminar discussion. I think anybody who's even not in the class is welcome to join, and we'll see you guys at like 5:05-ish for a break to get some water.
Thanks, everyone.
See you soon.
See you soon.
Thank you, Dave. Oh, hey, Dave, I have one more question